the human nervous system. You could argue it's the greatest mystery we have inside our own bodies.
Absolutely.
By weight, it's one of the smallest organ systems, but the complexity is it's unmatched.
Right.
We tend to make this lazy comparison to a computer, but that really falls apart, doesn't it? I mean, this system can literally rewire itself.
It can. It can create new pathways, modify its own structure from moment to moment. And that ability, that is learning. It's something no machine we've built can truly replicate.
It's the ultimate adaptive processor.
It really is. And when you look at how the body maintains balance, the nervous system is all about immediate control. It's about speed.
Milliseconds.
Responses are swift, they're brief, and it's a perfect contrast to, say, the endocrine system, which uses hormones that are slow but have effects that can last for hours or even days.
Okay, so let's unpack this. We are going to zoom in today, way past the brain and spinal cord, right down to the cellular level.
Fundamentals.
Our mission for this deep dive is to break down the anatomy and the function of the neural tissue, how it's organized, what the cells do, and how they talk to each other.
The blueprint of communication.
Let's start with that blueprint, the big picture.
Okay, so to start, the whole system is organized anatomically into two main subdivisions. First up, you have the central nervous system, or the CNS.
That's your command and control center.
Exactly, the brain and the spinal cord.
And the CNS is where all the heavy lifting happens, integration, coordinating sensory data with motor commands, and of course all the higher functions, intelligence, memory, emotion.
And it's important to remember it's protected. It's not just floating in there.
Right, not just by bone.
Right.
Mm-hmm.
The CNS actually developed from a hollow tube, and those spaces stick around in adults. They're the ventricles in the brain and the central canal in the spinal cord.
And they're filled with fluid.
Yep, cerebrospinal fluid, or CSF. It's a fantastic cushion, but it's also a transport medium.
So outside that protected zone, we have the peripheral nervous system, the PNS.
And that's pretty much all the other neural tissue. It's the link between the CNS and the rest of the body.
So if the CNS is mission control, the PNS is like the millions of cables running out to every sensor and every device.
That's a great way to put it. And functionally, the PNS is split based on the direction the information is flowing.
Okay.
You have the afferent division, which is all about sensory information. It brings information to the CNS.
And that information starts at receptors, right?
Right. They can be dendrites or whole sense organs or even specialized cells. And we group those receptors.
So you've got your somatic sensory receptors, which are what? Skin, muscles, joints, monitoring the outside world.
Correct. And then you have visceral sensory receptors, which monitor your internal organs, your digestive system, your respiratory system, that sort of thing.
Okay, so once the CNS gets that information and processes it, the signal has to go back out.
And that's the efferent division. Think of it as the motor freeway out. It carries commands from the CNS to the things that do the work, the effectors.
Which are basically muscles or glands.
Muscles or glands. And that efferent division is split again, and this split really defines conscious control.
Okay, so if I decide to move my hand, that's the somatic nervous system, the SNS.
Exactly. It controls your skeletal muscles. It can be voluntary, like moving your hand, or it can be an involuntary reflex, like pulling away from something hot.
But all the background stuff, heart rate, digestion, that's different.
Totally different. That's the Autonomic Nervous System, or ANS. Sometimes it's called the visceral motor system, and it's almost always happening outside of your conscious awareness. It's running your life support systems.
This seems like a good point to get some terminology straight because the names for structures change depending on if they're in the CNS or the PNS.
They do. And because the CNS is so heavily protected. So in the PNS, if you have a cluster of neuron cell bodies, we call that a ganglion.
Okay, and ganglion.
And the bundles of axons running together, those are nerves.
So a nerve is in my arm.
Right. Now, step inside the CNS. That same cluster of cell bodies is called a center. Or, if it has really distinct boundaries, we call it nucleus.
And the bundled axons.
Inside the CNS, they're called tracts. If you have a bunch of tracts running together, they form a column. So a nerve is in the periphery, a tract is in the spinal cord.
It seems complicated, but it's just a location marker.
It is. And one last one. The surface layer of gray matter in the brain, where so much processing happens, that's the neural cortex.
All right, let's dive even deeper now into the tissue itself. You have two main types of cells.
That's right. You have the communicators, the neurons, which are for processing and transferring information.
Yeah.
And then you have all the support cells, the neuroglia.
And this is where the numbers get wild.
They really do. The neuroglia, the support cells, outnumber the neurons about 5 to 1.
Which tells you just how much support these neurons actually need.
It's incredible. They need constant metabolic, structural, and protective support just to do their job. And it's critical because neurons are, for the most part, irreplaceable.
Right, because mature neurons don't have a centrosome, so they can't divide. But the neuroglia can.
The neuroglia can and do. So let's look at a typical neuron, a multipolar one. It all starts with the cell body, or soma.
The factory.
The metabolic factory, exactly. And a key feature you can see under a microscope are these clumps called Nissl bodies.
They're not just for decoration.
Not at all. They're clusters of rough ER and ribosomes, which means the neuron has a massive capacity for making proteins. It has to maintain its whole structure, especially that long axon. And those Nissl bodies are what give gray matter its color.
And coming off the soma are the antennae, the dendrites.
Highly branched, and they're covered in these tiny little projections called dendritic spines.
And that's where all the input comes in.
Up to 90% of the neuron's receptive surface area is on those spines. They're built to listen. The signal then leaves via a single long projection, the axon.
Which connects at the axon hillock.
The axon hillock, which is where the electrical decision to fire is made. And that axon can be incredibly long.
Like from your spine to your big toe?
Exactly. So the cell body needs a delivery system, and that's axoplasmic transport. It's an active process. It burns ATP to move materials down the axon to the terminals and bring waste back up. It's like a tiny biological highway.
Now, we classify these neurons structurally, right?
We do. The multipolar neurons, many dendrites, one axon is the most common. All your skeletal muscle motor neurons look like this.
Then you have the pseudo-unipolar neuron. It looks like a T.
It does. The dendrite and axon are basically one continuous process, and the cell body just hangs off the side. That's your typical sensory neuron.
And then there are a few rare types.
A few. You have bipolar neurons with one dendrite and one axon, which you find in sight and smell, and anaxonic neurons, which are small, and it's hard to even tell dendrites from axons.
Functionally, it's a bit simpler. It's about the job they do.
Right. Sensory neurons they deliver information to the CNS, and they rely on specialized receptors to do that.
So let's break those down. Exteroceptors are for the outside world, so touch, temperature, sight.
Correct. Proprioceptors are your sense of position. They tell your brain where your limbs are without you having to look.
And interoceptors monitor everything on the inside. Pressure, pain, taste.
All the internal systems. Now, on the motor side, you have the motor neurons, the efferent pathway.
And we touched on this. Somatic motor neurons go directly to skeletal muscle.
But the visceral motor neurons, the ANS ones, are more complex. They use a two-neuron chain to reach their target.
And connecting everything, the real decision makers.
The inner neurons. They are found only in the CNS, and their job is to analyze sensory input and coordinate motor output. They are the integrators, and they outnumber all the other neurons combined.
Okay, let's talk about that supporting cast, the neuroglia.
This is what's so fascinating. We used to think of them as just nerve glue, but they do so much more. Their jobs are dynamic and absolutely critical, especially in the CNS.
Let's start with the big ones in the CNS, the astrocytes.
The Swiss Army knives of the CNS. They have at least 4 major jobs.
Okay, what are they?
1, they control the chemical environment around the neurons, cleaning up excess ions and neurotransmitters. 2, they provide a structural framework. 3, they form scar tissue after an injury.
And the fourth one is maybe the most important.
I'd say so. They maintain the blood-brain barrier, the BBB. Astrocytes wrap these little cytoplasmic extensions, or feet, around the capillaries in the brain.
And that seals it off.
It creates an incredibly tight barrier. It isolates the CNS from anything harmful that might be circulating in your blood. And when astrocyte function gets altered, that barrier can break down, which we see in things like stroke.
Okay, next up, oligodendrocytes.
These are the myelin makers of the CNS. Myelination is just wrapping the axon in a fatty, lipid-rich membrane. It's an insulator.
And that insulation looks white.
It does. That's why we call areas with lots of myelinated axons white matter. The insulated segments are internodes, and they're separated by these tiny gaps, the myelin sheath gaps, or nodes of Ranvier.
The security force of the CNS.
That would be the microglia. They're the smallest glial cells, and they are phagocytic. Basically, they're the resident immune cells eating up debris, waste, and any pathogens that get through.
And their numbers shoot up during an infection.
Dramatically. Finally, you have the ependymal cells. They line the central canal and ventricles and help produce and circulate the cerebrospinal fluid.
And in the PNS, it's much simpler. Just 2 types.
Just 2. Satellite cells surround the neuron cell bodies in the ganglia, regulating their environment.
And then the famous Schwann cells.
Schwann cells cover all peripheral axons. In myelinated axons, a single Schwann cell wraps itself again and again around one segment of one axon, forming that insulating sheath. The outermost layer with the nucleus is called the neural lemma.
And this foundation helps us understand some clinical issues, right? Like pins and needles.
Yes, that's called paresthesia. It's that numbness or tingling you get from sensory nerve damage. It can be temporary, like when your arm falls asleep, or it can be permanent.
And even something as common as a headache has different origins here.
Exactly. A tension headache might be muscular, but a migraine is a serious neurological and cardiovascular event tied directly to these structures we're talking about.
Okay, we have the cells, we have the support. How do they actually send a signal?
It all comes down to excitability. That's the ability of the neuron membrane to respond to a stimulus. And if the stimulus is strong enough,
You get an action potential.
you get the all-or-nothing electrical event, the action potential. It's a rapid change in potential that travels or propagates down the axon.
And here's where it gets really interesting. The speed of that signal is not the same for every neuron.
Not at all. Two key factors control the speed: myelination and diameter.
Myelination first.
Myelinated axons are way faster, 5 to 7 times faster than unmyelinated ones. The myelin sheath forces the electrical current to jump from one node of Ranvier to the next. It's called saltatory conduction, and it is incredibly efficient.
And then there's the diameter of the axon.
Bigger is faster. The largest myelinated fibers in your body are like a high-speed rail network. They can move signals at up to 140 meters per second.
Which is hundreds of miles an hour.
It is. Compare that to the smallest unmyelinated fibers that carry some pain signals. They travel at less than 1 meter per second. The system prioritizes information based on speed.
But for all this incredible design, there's a huge downside. Recovery from injury is limited.
Extremely limited.
Right.
In the PNS, if an axon is damaged, the part of the axon after the injury dies off, that's called Wallerian degeneration. Macrophages come in and clean up the debris.
But there's a chance of recovery there.
A chance, yes. The Schwann cells are the key. They divide and form a sort of tube, a guide, and they release growth factors. If the axon sprout can follow that guide, it might reconnect, but it's often incomplete.
And in the CNS, it's a different story.
A much bleaker story. In the CNS, repair is almost impossible. You have more axons involved, and the astrocytes, which are normally helpful, form scar tissue that blocks regrowth.
And they actually release chemicals that stop it.
They do. The environment is actively hostile to repair. It's why spinal cord injuries are so devastating.
All right, the final piece of the puzzle. How do these neurons talk to each other?
At the synapse. That's the junction where a neuron communicates with another cell.
And the most common type is the chemical or vesicular synapse.
Right. It uses chemicals called neurotransmitters like acetylcholine. And because only the presynaptic side releases the chemical, the communication is strictly one way.
And it's a very precise process.
A four-step process. First, the action potential arrives and triggers the release of the neurotransmitter. Second, it diffuses across the gap and binds to receptors on the other side.
And that binding causes a change.
It does. Step 3 is that the binding changes the permeability of the postsynaptic membrane. This can cause either excitation, which encourages a new action potential, or inhibition, which makes one less likely.
And then it's over quickly.
Very quickly. The neurotransmitter is either broken down by enzymes or reabsorbed. The signal is brief.
Then you have the really rare ones, the non-vesicular synapses.
The electrical synapses. Here, the cells are physically connected. Ions pass directly from one cell to the next. It's incredibly fast and often bidirectional.
So what makes a neuron actually decide to fire?
It's all about integration. You have to remember, a single neuron is getting input from thousands of other synapses. Some are telling it to fire, some are telling it not to.
It's a constant negotiation.
A constant negotiation, and the final decision is made by summing up all those signals at the axon hillock. If the total excitatory push wins, it fires.
And these neurons don't just work alone, they're organized into neuronal pools.
And within those pools, the wiring patterns, or circuits, determine how information is processed.
There are 5 basic types.
First, divergence. That's where information from one neuron spreads out to many others. It's for broad distribution, like a single sensory input informing multiple brain regions at once.
And the opposite is convergence.
Where several neurons all talk to one single neuron. This allows for very complex control. Think about breathing. You can control it voluntarily, but your brainstem is also controlling it involuntarily based on CO2 levels. Those signals have to converge on the same motor neurons.
Then you have serial processing, which is just a simple step-by-step relay.
Simple chain of command. Parallel processing is much more dynamic. Multiple pools process the same information at the same time.
This is why if you step on a nail,
Exactly. You withdraw your foot, you shift your weight to the other foot, you feel the pain, and you shout, ouch, all at the same instant. That's parallel processing,
and the last one is reverberation.
which is a positive feedback loop. Collateral branches of an axon circle back and re-stimulate earlier neurons in the chain. This keeps a circuit active, and we think it's important for things like consciousness and muscle coordination.
So to wrap this up, what does this all mean? We've seen this incredible organization from the glial cells forming the blood-brain barrier all the way to the different speeds for different signals.
Right. And the central idea isn't just about one action potential. It's about the complex integration, the constant summation of excitatory and inhibitory signals that allows the system to function.
We started out by comparing it to a computer, but the reality is the complexity of those neuronal pools with convergence, divergence, reverberation.
It's a level of processing capability that is just, well, it's biological. It's unmatched by anything we've engineered.
Okay, here's a final thought for you to chew on. We talked about how the most common synapses are chemical, which keeps the communication brief and one way. What would happen to your perception or your consciousness if those rare bidirectional electrical synapses or those positive feedback reverberation circuits suddenly became the dominant way your entire cortex communicated?
A network that could never stop firing.
Something to think about.